Hello students, this is Dr. Faizan Mirza. I'll be discussing with you question 21 till question 40 of the paper October-November 2021, variant 11. So question 21, hello guys. So question 21 actually tells us that which statement helps to explain why two sugar phosphate chains in the DNA molecule are constant distances apart. So this is a fact because the purine binds with the pyrimidine. So because of the purine and pyrimidine bonding together, the width stays the same. So that's a fact. You just look for that, and that's the basic answer. We know that the purine are the larger bases. If two purines were added together, the DNA width would increase at that point. If two pyrimidines were added together, as complementary bases, then again that the DNA weight would decrease at that point. So question 22. During DNA replication, what must happen before a newly DNA nucleotide is bonded to the next nucleotide? We have three things happening: complementary base pairing, hydrogen bond formation, phosphodiester bond. We know whenever DNA is replicating or whenever mRNA is forming, first of all, complementary base pairing must occur by hydrogen bond formation, and phosphodiester bond is the last to form. Keep that in mind. Complementary base pairing occur first. It is stabilized by hydrogen bonding, and the reaction is carried by DNA polymerase in DNA and RNA polymerase in RNA. So the answer will be 1 and 2. 3 cannot be the answer. Question 23. An antibiotic enters the cell through a membrane channel protein. If it's entering through a membrane channel protein, that means that the antibiotic must be a polar molecule. If it's a polar molecule, it must be a hydrophilic molecule. It must be a molecule that only can enter through channel protein. That's already mentioned to us. Some bacterial cells have shown resistance to this antibiotic by acquiring a mutation, which means there's a gain of function, which alters P, which is the channel protein. This mutation prevents the entry of antibiotic into the cell, so the antibiotic cannot enter the cell anymore because of this mutation. Which conclusions can be drawn about how the resistance to this antibiotic developed? We know that antibiotic resistance develops by mutation, and mutations occur by chance. So out of these three statements, the mutation changed the order of amino acids. Yes, this might be correct. In the gene coding for P, no. Whenever mutation occurs, yes, the amino acid changes occur, but in the gene, the change is of nucleotides, not the amino acids. The amino acids change in the protein and the nucleotide change in gene, so the statement is wrong. Mutation results in production of P with altered tertiary structure, 3D structure change. So yes, it will not allow the same molecule to cross through. The antibiotic is hydrophobic. No, we already have seen that it cannot be hydrophobic because it's using a channel protein to enter. So only the option 2 is correct. Again, you can see they have given 3 in 3 options to just confuse you that 3 must be there as an answer. Again, in this case, it's not so. So always use a pencil and keep on just annotating with the question. Don't just keep on solving things orally. Always annotate your question. You are allowed to use a pencil on the question paper. You can write the description, whatever hints you want to write down, which can help you in which can assist you in coming to the right answer. Question 24. What is the correct tRNA anticodon for the amino acid proline? Pure proline DNA triplet is given to as GGT. We know that if the DNA codon is GGT, its mRNA would be CCA. C against G, the C against another G, and against this T you will have A. These are the mRNA codons. But since they are asking you the tRNA, so we'll convert this mRNA into tRNA. So this becomes GGU. So that's how the template stranded tRNA comes. A shortcut, if you can just keep in mind, if a DNA codon is shown to you and you are asked for the tRNA, the DNA codon will be same as the tRNA. The only difference will be between thymine and uracil. So let's move on to question 25. The question 25 here is actually they have said that the table shows the observation made by students who treated 3 different types of cells, P, Q, and R. Transfer section of plant stem is shown to you using a light microscope. See, 3 cells are shown, P, Q, and R. You can probably use the links mentioned for the ratio of cell wall width to the whole cell. What I preferred, I just use a structure observed of cell in cell contents to arrive at the answer. My answer gives me options of companion cell, xylem element, and phloem sieve element. So these are the 3 type of cells given to me in my options: companion or xylem, phloem or companion or phloem, xylem vessel, and again the same ones here. So if I'm seeing through a cell P and I see nothing inside the cell, that must be xylem. So answer should be either C or D. It cannot be a companion cell. Now the cell Q is having regular pattern of circles, and the circles in some cells must be sieve pores in the sieve plate. So yes, Q should be the phloem sieve element. Answers cannot be C. And coming to the last, the granular appearance of all cells will be because of the cytoplasm and the extensive organelles which are present here. So this must be the companion cell. The answer becomes D. Water molecules are attracted to each other. If they are attracted to each other, which property of molecules is important for upward movement in xylem? We know water molecules are attached to each other by hydrogen bonding by cohesion. This is just a factual question, asking you to recall cohesion. Addition is between water molecules and the vessel wall. The diagram shows model to demonstrate the mass flow. You have two beakers. Inside the beaker you have water. You can see water is moving, and if water is moving, then X must be xylem. So if that's the condition, we know that if X is xylem, so answer must be either A or B. Now there's concentrated sucrose solution here, concentrated sucrose solution here, and it's moving in either direction, either this direction or that direction, and it's moving across phloem from phloem. So phloem must be W. So our answer should be either A or B. It cannot be C. Now the next thing is concentrated sucrose solution is mentioned to you. We know the solution concentrated will always be in the source cell, which is leaf. So our Y should be leaves. And similarly, dilute sucrose solution should be in the sink, and this is in the roots. So yeah, the answer becomes roots. Sorry, the leaves are Y and the roots are Z. In this case, the direction is from Y to Z. And I hope that explanation of how concentrated sucrose solution, this is the actual hint that you have to catch for the dilute solution and the concentrated solution, which is in the sink, and the sucrose accordingly, and sucrose moving in the phloem and water moving in the xylem. So answer becomes B. The parts of heart control, heart action are listed. You are given SAN, AVN, and Purkinje tissue. Atrial contraction is not by AVN, it's not by Purkinje, it's not by SAN and AVN combined, it's only by SAN. SAN and AVN do not combinedly cause atrial contraction. Atrial contraction is only because of SNO. Ventricular contraction is because of Purkinje. Again, you just cross out the wrong options. And ventricular contraction, the excitation is produced by SAN. Yes, this option is correct, but the option given against atrial contraction is wrong. So other options are just outright wrong. The diagram show part of circulatory system in mammals. You know, if this is a branch structure shown to you, this must be capillary. These must be venule. These must be small veins, and this is the vein actually leading to vena cava eventually. Where is the blood pressure and the blood flow lowest? So keep the fact in mind, blood pressure is least in the veins in our circulatory system. Actually, it's least in the right atria, but in the vessel, it is least in the vein. And the lowest speed of the blood flow is in capillary. So answer becomes D. Photomicrograph of human blood cells are shown to you. You can see, just annotate the diagram. All these are your red blood cells. These large cells are monocytes. You can see this indented nucleus inside. So these must be monocyte. This is a neutrophil. How do we know it's a neutrophil? Because it's having a lobe nucleus. You can see that the nucleus is lobe. And if it's a lobe nucleus, this must be a phagocyte. A neutrophil is a phagocyte, we know. Monocyte is also a phagocyte. So what type of blood cell is X? Is it a B lymphocyte? It's a monocyte. Is it a T lymphocyte? No, it's just a phagocyte. The diagram shows some structural reactions, some reactions when when carbon dioxide enters in blood from cells in the metabolically active tissue. Which reactions catalyzed by enzyme carbonic anhydrase? Always carbonic anhydrase is present inside the cytoplasm. So A and B cannot be answered because they are given in plasma. So they are wrong. The answers could be either C or D. We know that the carbonic anhydrase catalyzes the reaction forward of CO2 and water combining to form H2CO3, and the same reaction reverse can also be catalyzed by breaking H2CO2 into carbon dioxide and water. So in this case, since the blood cell is just passed from the metabolically active tissue, the answer is C. The graph shows you oxygen dissolution curve of hemoglobin and animals that live at high altitude and the animals that live at low altitude. This is high altitude, this is low altitude. This y-axis shows you partial saturation of hemoglobin with oxygen. X-axis shows you partial pressure oxygen in kilopascals. As you increase the partial pressure of oxygen, the percentage saturation of hemoglobin is increasing. Now, whenever such curves are given to you, there's a basic fact that I ask my students to just keep in mind. If the curve is closer to y-axis, you can see this curve is quite close to y-axis. If this curve in hemoglobin saturation, or you can say the dissociation curve is closer to y-axis, this curve being closer to y-axis shows to you a very high affinity of that hemoglobin with oxygen. And high affinity for oxygen means that oxygen will not be released readily. Whenever you have these curves, for example, if I had another curve here, another curve here, so I would mention that this curve, which is closest to y, it is having the highest affinity for oxygen. It would not release oxygen readily. And this reverse argument goes for any curve which is shown to me furthermost from the y-axis. The reverse argument, it would have the least affinity and will readily or release oxygen very quickly. So if we read the statement, hemoglobin is having a higher affinity of oxygen. Look for the curve close to y-axis. Yes, A is correct. Hemoglobin releases oxygen more readily for high altitude. No, this is exactly in opposite. The change in partial pressure of carbon dioxide causes Bohr's effect. This answer might just be correct, but there is no reference of carbon dioxide mentioned in the entire graph or the question. There is no concentration shown to you for carbon dioxide, no color pascals given. So this can't be an answer. The decrease in percentage carbon dioxide, again, carbon dioxide, since it is not mentioned in any of the information given to us, answer C and D just cannot be there. Answer B is actually a wrong science. So we are left with answer A and the logic I just explained to you. The diagram shows three features of found in mammalian gas exchange system. Ciliate epithelium, goblet cell, cartilage. Just recall trachea and bronchus only are the structures in the gas exchange system that have all the three structures: ciliated epithelium, goblet cells, as well as the cartilage. Bronchioles lack cartilage. Bronchioles will have ciliated epithelium, they may have goblet cells, but they will not have the cartilage. They just cannot have cartilage. So we just cancel out the bronchioles. Yes, trachea is correct, but bronchus is not mentioned, so that's correct, incorrect as well. We are left with trachea and bronchus. Short-term effects of nicotine are not in syllabus, so you can ignore this question. Question 35, some features of the disease are listed. You need to be very careful when identifying the disease here. It can be transmitted by animals to other animals, including humans. So that means their insect vector is involved. It might be mosquito causing malaria, or it might be TB. One mode of transmission is by transfusion with contaminated blood. So contaminated blood can be because of can cause HIV, TB. Yes, malaria can also be transmitted by contaminated blood transfusion. The causative organism can show multiple drug resistance. We know MDR TB and malaria has multi-drug resistance. The falciparum plasmodium of malaria is drug resistant, and multiple drug resistance is shown by them. The majority of humans who die from disease are children. This fact is not for TB. TB is fatal, yes, but it will not kill children, because by the time if a child is having TB, by the time the child actually dies from the disease, the child will be a grown up, because TB is a very slow infection. It is a very slow infection, and yes, it is fatal if it's not treated properly, but this option is wrong. And with this, if we just arrive at the answer, so we know cholera, HIV, tuberculosis cannot be our answer. Answer will be malaria. The following advice was given to a person traveling to a country where there has been an outbreak of infectious disease. All the information given to us highlights cholera, because it's all the oral route, how the disease is going from the oral route to the body. So that must be cholera. So that's a straightforward question. Which description of multiple drug resistance is bacteria is correct? If it's multiple drug resistant, that means the bacteria is having genes for various antibiotics. So bacteria have DNA with resistant genes for several different antibiotics. Yes, that's correct. Different species of bacteria have DNA with resistant gene for one type of antibiotic. No, this is the wrong thing. Always annotate what's wrong. Always mention with the pencil that no, this is wrong. So you can narrow down your options for the correct answer. Larger of copies of one species of bacteria are immune to several type of antibiotic. Many different species of bacteria are immune to one type of antibiotic. You can never use the term immune for bacteria. A bacteria cannot become immune. Bacteria do not have immune system. Immune system is primarily in mammals, and more importantly in humans, it's most evolved. So we do not use the term of immune system or immunity or immune for bacteria. What's the description for T lymphocyte? T lymphocytes are only found in blood. No, they are found in blood, tissue, fluid, and lymph as well. So A is wrong. They can leave blood, accumulated site of inflammation. Yes, they can leave blood and secrete cytotoxin. No, cytotoxins are released by killer T cells only. And when they are exposed to bacteria, no, when they are exposed to the antigen presenting cells. They circulate in blood, are always present antigen. No, they do not present antigen. The antigens are presented by either antigen presenting cells, which are monocytes, macrophages, or infected cells of the body. Question 39. Where are antibodies found during an immune response? They are found on the surface of memory cell. Yes, they are found in blood plasma. That's very correct. So 39 is straightforward. Question 40. Monoclonal antibodies are used in diagnosis and treatment of disease. They are produced using a technique known as cell fusion. Which two cell structures are fused? We know cancer cells and plasma cells are fused together to get this habitoma cells. So that was all about the question paper of October, November 2021, variant 11. I hope that was helpful for you guys. That's all from my side. Thank you so much.